Hey guys, welcome to Biophysics class 36, and this class is on the Hodgkin-Huxley model. This is our final model, our culminating model of the action potential, which we've been studying for several classes using first the Donnan equilibrium, then the GHK equation, and then some circuit ideas of circuit models of the action potential. But the Hodgkin-Huxley model builds on the circuit model and also includes the ideas of the Nernst potential into a fully integrated model of the action potential. So first, a little bit of history and background. Hodgkin and Huxley were wanted to study the action potential in nerve cells, but didn't have equipment to electrodes small enough to study it in mammals and smaller organisms. So they looked at the giant squid, whose axon connecting neurons in the brain down to its tail, or its base here, are quite enormous, several centimeters in diameter. So the electrodes back in 1952, when Hodgkin and Huxley developed this model, were sufficient size to sufficiently precise to record action potentials and voltages across those axons. A second interesting aspect is that the model was developed and tested and refined before the use of computers. And you'll see when we get to the form of the equations that they're fairly involved differential equations, and this was done without the ability to, without knowledge of genetics or the ability to manipulate proteins and many of the tools of modern biology. So Hodgkin and Huxley were working with oscilloscopes and microscopes and electrodes and making guesses at what was going on. And they had to compute each time and see if it agreed with their readings. And all these computations were done by hand. So it was quite an amazing accomplishment, and was rewarded in 1963, 11 years later, with the Nobel Prize in Physiology and Medicine. And amazingly, after over 60 years, or almost 60 years, it remains the most widely used model for the action potential. Okay, so it's a very it's amazing what Hodgkin and Huxley were able to do back at such an early stage in the revolution in biology. So how does it work? Well, we start with our electric circuit model, where the cell membrane, or the axon membrane, is modeled as a capacitor separating charge on either side of the membrane, ionic charge, and where the ion channels are modeled as conductances or resistors, and where the Nernst potential, the tendency of ions to move back and forth, is modeled as a battery, okay, or as a battery. So so we have this model then for our basic squid axon and most neurons. We have sodium and potassium and what's called a leak current. And a leak current is all the gates, the current through all the gates, which are always open. The sodium and potassium gates are, most of them are voltage gated or membrane potential gated. They change their conductivity with the membrane potential V, but the leak gates are always open. So what goes through the leak gates, well, it would include some sodium and some potassium, but also other ions, calcium and chlorine, and several others. So it's everything that isn't involved in the voltage-gated sodium and potassium. And then we have our capacitor and our three Nernst potentials acting as driving forces to move the sodium and potassium and leak currents through the membrane. So quantitatively, our total current will be our potassium plus our sodium plus our leak current. And each of these can be written as a function of the membrane potential and time, and is equal to the difference between the Nernst potential and the membrane potential. Because remember, if the Nernst potential is equal to the membrane potential, no current of that ion will flow. That ion has competing tendencies which are satisfied by a membrane potential equal to its Nernst potential. So this is the effective potential for sodium times the conductivity of sodium, which also depends on voltage and time. And we have similar expressions for potassium with the effective driving potential of potassium times its conductivity, and the leak, which is the leak membrane, the composite leak Nernst potential over all the involved ions minus the membrane potential, times the conductivity. And notice the conductivity doesn't depend for the leak current on voltage, because the leak gates are not voltage dependent. They're not voltage gated. Okay, so let's dig in a little bit more to the form of the conductivities, the conductances. The conductances take the form in the model, the Hodgkin-Huxley model, of a maximum value times a fraction of channels which are open, fraction f of channels which are open. So if all of the, for example, all of the sodium channels are open, we're at a membrane potential where that's the case, where F would be equal to 1, then that we would be at maximum conductance for sodium. On the other hand, if none of the channels were open, we were at a membrane potential where none were open, we would have 0 conductance. And G max F, this fraction, would be between 0 and 1. So G max would be the maximum value associated where F, with the case where F was equal to 1. So that's going to be the form that we use for the conductance, a maximum value times the fraction of channels which are open. To wrap up this differential equation, we need another form for the current, and that would be equal to the charge on one side, the derivative of that charge, which, because it's a capacitor, is going to be equal to Q is equal to CV. dQ dt is C dV dt for capacitor. So we can write then the current is equal to C dV dt, which is equal to the sum of these three currents. And so we have C dV dt is equal to Vk minus V, and so forth. Okay, so this is our differential equation for the membrane potential V, and we know that we have, in these conductances, we have more voltage dependence, and we know that it has to do with the fraction of open channels. So let's look at that in a little bit more detail. So the key concept in this is called a gating variable. Each of the gates, the sodium and potassium gates, can be closed or open, and they can go back and forth between being closed and open, depending on the membrane potential and time. So we call a gating variable F the portion of channels of a particular type which are open. So if F was equal to a half of the, for sodium, half of the sodium channels would be open and the other half would be closed. So if so F is the fraction of open channels, also known as the gating variable. If F, since a channel is either open or closed, if a fraction F is open, then 1 minus F will be closed. So if 25.25 fraction of sodium channels are open, 0.75 would be closed, 1 minus 0.25. All right, so how can this fraction change? Well, there's two ways. We can have a closed channel open, or we can have an open channel close. So for the first process, the rate of the change in the portion of open channels would be equal to the portion of closed channels times the rate at which closed channels open, which is called alpha. Okay, so if we take the fraction of closed channels times the rate at which closed channels open, that's going to increase the number of open channels. We subtract off the fraction of open channels times the rate at which open channels close, because that's going to decrease the fraction of open channels. So beta here is the rate at which open channels close, which depends on V, as does alpha. They're both voltage dependent. Okay, so we're going to have gating variables associated with potassium activation, sodium activation, and sodium inactivation. And instead of F, to distinguish them, they're going to be called N for potassium activation, M for sodium activation, and H for sodium inactivation. All right, so let's start off with potassium activation, or GK, as a function of V and T. We can write this as GK max, or GK bar, times a gating variable N to the fourth power. So GK bar is the maximum conductivity with all gates open, and our gating variable N is equal to 1. And then you'll notice this is to the fourth power, okay? And that seems really curious, but that's because each potassium channel has actually 4 activation gates. There are 4 gates. So because the 4 gates are open or closed independently of each other, to get the chance of all of the gates being open, we have to take the chance of each of the gates being open multiplied all together. So it'd be n times n times n times n, or n to the fourth. And this is a picture of a potassium gate, or a diagram with the proteins drawn schematically. And it has 4 tetramers here in a ring shape. And all 4 of these have to be in an open position for the ion to pass through, which explains the fourth power here. Okay, and then so we have the fourth power because of the 4 gates in the channel, but then the gating variable N follows the form we saw in the previous slide for F. The change in the number of open potassium channels is equal to the fraction of closed channels times the rate at which closed become open, which is alpha, minus the fraction of open channels times the rate at which open become closed, which is beta. So again, same as the previous slide, but where it was F, but it's now called N for potassium activation. And as was mentioned, alpha and beta depend on voltage, on membrane potential, and those functions are chosen so that the potassium gates begin to activate and deactivate at the right points in the action potential. So for potassium, that's around 0. It starts to activate, so potassium will flow out on the downstroke, and it begins to deactivate at or below minus 20, so the potassium will stop flowing out and come to the resting potential. Okay, let's move on to sodium current activation, GNA via T. And we actually write this, the sodium conductance, as a maximum sodium conductance times the product of two terms. The first one is activation, the M term. The second one is inactivation, which we'll deal with in a minute. So for the M term, it's M cubed for the same reason that potassium was N to the fourth. There are actually 3 activation gates in the sodium channel, resulting in the third power for the gating variable M, the activation variables for sodium. So here are the 3 gates, which are activation variables. The other gate here is an inactivation gate, which is associated with the variable H. And then with the first, for the activation variable, we have the same form of equation. One more time, the change in the number in the fraction of open gates is the fraction of closed gates times the rate at which closed become open, minus the fraction of open gates times the rate at which open become closed. Okay, so hopefully that's starting to sink in and become familiar. And as in potassium, for sodium, the rates at which open become closed, closed become open, and open become closed as a function of membrane potential are chosen so that the sodium gates begin to activate at the right time and deactivate at the right time. For sodium, that's about minus 50 millivolts, so that it rushes in, creating the upstroke, begins to deactivate at or below minus 30 millivolts. So it's gone on the, so it's lessened on the downstroke. Finally, we get to the inactivation sodium current, where we have the H variable. Notice that inactivation means no current will flow. So that would correspond to the value H equals 0. Inactivation gate would be, it would be inactivated, would correspond to an H value of 0, giving a conductance and therefore a current of 0. There's one inactivation gate, as we mentioned. So this H is to the first power, and it obeys a similar equation as the activation for sodium and potassium. And the rates at which the closed goes to open and open goes to closed are chosen for sodium gates to slowly inactivate around minus 20. Okay, so around here, and then very slowly begin to de-inactivate down around minus 40. That prevents sodium from rushing in during the downstroke and allows the potassium to rush out and decrease the potential back down to its resting potential. Okay, so at this point, we're ready to put together the full Hodgkin-Huxley model. Again, it's based on a couple of ideas. The idea of the circuit model. And the circuit model has, again, conductances for potassium and sodium and the leak current with their Nernst potentials driving each, and the cell membrane as a capacitance. So we have this equation, which is for the membrane potential. The first equality is from the capacitance, the second from adding up the three currents, which are the effective potentials times the conductances of each. And now we know that the conductances are voltage and time dependent. And for the potassium, we know that it can be expressed as a gating variable, as a maximum conductance times a gating variable n to the fourth power, because there are 4 gates in each channel. For sodium, there are 3 activation gates, so we have a third power, and 1 inactivation gate, so we have a first power of the other variable h, and the leak current is just always has the same conductance. It's not voltage dependent. For each of these gating variables, we have n, m, and h. We have equations which represent the transfer of gates from open to closed states, and the rates at which those occur are the alpha and beta parameters. Beta being how open goes to close, and alpha how closed goes to open. So we have 4 variables, the membrane potential V, and the variables n, m, and h, which are the gating variables. The fraction of channels of potassium activation, sodium activation, and sodium inactivation. The parameters are the capacitance and the membrane potentials, the Nernst potentials, which depend on the concentrations of ions inside and outside the cell, as well as the maximum conductivity or conductance of each type of channel, which depends on the density of ion channels in the cell membrane or the axon membrane. And the 3, as we saw, the 3 alpha and beta functions, the rates of closed to open and open to closed and closed to open, are chosen so the channel gates open at the appropriate membrane potentials and with the appropriate speed to create the action potential for a particular type of neuron. So that's the Hodgkin-Huxley model. 2 basic ideas: the circuit model with the capacitor and the channels as wires with conductances, also known as resistances, and the idea of fractions of open gates and conversion between open and closed gates through these form of equations here. So I hope you appreciate the complexity of this model and can appreciate how difficult it might be to create this model before having computers to test out measurements, and what an accomplishment it was for Hodgkin and Huxley to create this back in the pre-computing era. So that's the Hodgkin-Huxley model, and I hope you have enjoyed this presentation, and I will see you in class.